Welcome back, biotechnology enthusiasts. Today, we are going to dive into the groundbreaking discovery that has taken the scientific world by storm. Synthetic biologists from Indiana University have created genetically minimal artificial cells, proving that when it comes to life, it finds a way. Join me as we explore the fascinating results of this experiment and what it means for the evolution of cellular complexity. Our story begins with evolutionary biologist J.T. Lennon and his team, who studied a synthetic minimal cell with 45% of its genes eliminated, reducing it to the smallest set of genes required for autonomous life. Despite its reduced genome, Lennon's team found that this minimal cell evolved as quickly as a regular cell, showing the inherent resilience of life. They discovered that a synthetic cell with a reduced genome could evolve as quickly as a normal cell. Despite losing 45% of its original genes, the cell adapted and demonstrated resilience in a laboratory experiment lasting 300 days, effectively showcasing that evolution occurs even under perceived limitations. Understanding how organisms with simplified genomes overcome evolutionary challenges has important implications for long-standing problems in biology, including the treatment of clinical pathogens, the persistence of host-associated endosymbionts, the refinement of engineered microorganisms, and the origin of life itself. This research done by Lennon and his team demonstrates the power of natural selection to rapidly optimize fitness in the simplest autonomous organism, with implications for the evolution of cellular complexity. In other words, it shows that life finds a way. So, what does this mean for the future of synthetic biology? Well, it opens up an array of possibilities for the development of new treatments for diseases and the creation of sustainable microbial ecosystems. With the understanding that even with the most minimal genes, life still finds a way, the potential for creating and engineering living organisms gets more exciting every day. Now, let's take a closer look at the science behind the experiment. Using a paper recently featured in the journal Nature as our guide, we'll explore the various stages of this remarkable study, from the creation of minimal cells to their evolution and adaptation. The power of minimalism. Scientific research has often been about expanding our knowledge by adding to it. But with this new experiment, biologists explored the limitations of life by taking away from it. Let's find out how scientists managed to create genetically minimal artificial cells. The evolutionary challenge. The experiment explored how a gene-reduced cell behaves in comparison to a regular cell. The experiment was executed with 300 days of adaptation, after which the results were astonishing. Let's learn more about the findings. The remarkable results. The experiment proved that genetic minimalism does not hinder the growth and evolution of an artificial cell. The survival and successful evolution of minimal cells have significant implications for a range of applications, from creating new treatments for diseases to developing sustainable microbial ecosystems. What does this mean for our future? Life finds a way. One of the most significant findings is how, despite the 45% reduction of original genes, the minimal cell adapted and demonstrated resilience. Even with such limitations, life finds a way. Let's appreciate the resilience of life. Exciting future of synthetic biology. The results of this experiment in synthetic biology are truly thrilling. The possibilities for innovative treatments and sustainable systems are vast. With this knowledge, biotechnologists have a better understanding of the resilience of life and the power of evolution. Are you as excited as I am?